Okay, thank you to all of you for being here. Okay, my voice is not at its best, but please, if you haven't downloaded, go to the QR code that you'll find on the screen because we are going to have an interactive section here within this lecture just to make sure that all of you be part of the presentation, okay? So download the QR so that way you'll be interacting with us along the presentation.
So while you are downloading the program, I will do a brief introduction. It's a pleasure to be here today. I guess it's been 6 years since Dr. Nell and myself came to Columbia to initiate this Botrytis and thrips research program. And so I've been traveling here for several years and see several familiar faces in the audience. It's been a pleasure working with the Columbian growers on this project. Some of you were at Seafloor a couple weeks ago, and we presented some of our other project that we are doing with the American Floral Endowment that is focused on chrysanthemum leaf necrosis.
Okay, do all of you have the app already? Yeah.
So all of the work that we are presenting today is basically Melissa's graduate career. She did her master's degree and PhD in my lab over the last 6 years. And I think she's responsible for all of the data that we're showing today. So, and we would like to also, as we get started, we have to thank some of the growers that we've worked with here in country. These are experiments that we cannot do ourselves at my university very easily because we cannot grow roses in South Carolina so well. And so we require the cooperation of the Columbian growers to actually do treatments in their greenhouses here. And then we would ship flowers to my laboratory where we could evaluate them. So this project, the success of this project is highly dependent on the cooperation that we've gotten, most specifically from the GHT group, the Ultra Golf Farm, and Farms and Sunshine Bouquets. So the outline for today's presentation will go as follows. And as we go through the slides, we will ask you questions. We have about 20 questions that we hopefully you find to be fun and interesting way to be engaged in the presentation. And so we will ask you a question about your opinion and knowledge about Botrytis, and you get to vote on your phone, and then we'll show you what we believe to be the correct answer. Sometimes these things are a matter of opinion. Mostly they're based on data that we've collected. But sometimes there's opinion involved, so we'll share with you our opinions. And then at the end of the presentation, we have awards and prizes, very small prizes, for the first and second place, people that have had to get the most questions correct. Okay, and keep in mind you only get when you enter your answer, it's final. You cannot change your answer once it's been entered. Okay. All right. So the outline for today's presentation, we'll talk about the biology of Botrytis, the cycle of life, symptoms that indicate Botrytis infection on cut flower crops, the dispersion and susceptibility of the dispersion of spores and the susceptibility of plants to fungal infections or Botrytis infections, how best to monitor for Botrytis, cultural practices for Botrytis management, what environmental conditions are ideal for Botrytis growth, how to manage fungicide application and fungicide resistance. And then we have presented a lot of this information at previous meetings and through newsletters over the past several years, so some of it will be familiar to you. The last part, where we talk about alternative strategies to traditional, conventional fungicides, Melissa will show you some of her newest research that should be new information for everybody in the audience. Okay, so when we talk about diseases, We like to have the disease triangle as our reference point, because to have a pathogen infect our plants, you have to have 3 simultaneous factors occur. And that being, you have to have the presence of the pathogen, which seems obvious. And with botrytis, this is a very difficult one to control because botrytis is everywhere. We try to limit the amount of inoculum, the number of spores, but this is a difficult management practice. In the lower left-hand corner, you have to have the appropriate environment. And in North America, this is the piece of the triangle that we most often focus on, because we have heated greenhouses, and so with the heat at night, you can prevent condensation. And whereas in Colombia, it's much more difficult to prevent condensation. And so it's hard to manage the environment for botrytis here. It's a very good environment for growing flowers and botrytis. And then you have to have a susceptible host, meaning you have a crop that is susceptible, and for the most part, all of our cut flower crops are pretty highly susceptible. So we have a perfect storm for botrytis in this part of the world. Makes it a very big challenge. Our presentation will focus initially on the pathogen, and then we'll talk about the environment, and finally how can we improve host resistance to botrytis infection.
I'm sorry, before we start, just to keep everyone within the same context and making sure that we all have the same terminology, I am going to start with a little bit of the terminology, regarding botrytis. Botrytis is a fungus that is characterized because it reproduces basically in a sexual way through some spores that are called conidians, and those conidians are produced in an organism that is called the conidoforms. We also have some vegetative structures that belong to the fungus that are called hyphas, and we are going to know it as micelle. Additionally to that, when the spores they get to the plant, they are going to, and they start their infectious process, they are going to get a filament, and as the filament, they are going to create a filament that gets in contact with the flowers. And here it has another peculiar thing besides the reproduction through the conidians. It also has another inoculate structure that is called the sclerosis. And those are the resistant structures that remain on the debris and on the floor, on the soil. And that is something that analyzes and has high resistance to high temperature and UV light. So to start with, we are going to go to the pool, and we are going to make a trial here. So you're going to tell me, accordingly to the idea that you already had about the disease, how do you think is the chronological development of the life cycle of this disease? What is it? How does it develop? Okay, arrange the following steps. I am going to read the question in English for the ones that do not speak Spanish. For the English speakers.
We need to communicate,
Yeah. So the question is, please order the following steps for the life cycle of botrytis in a chronological manner. The first one is the arrival of spores and the recognition of the host. The second is the spore germination. The third is appresorium formation, then invasion of the tissue and mycelial growth, and sporulation.
Okay. Everybody have their answers entered? All right. then we will reveal the correct answers.
We will explain the exercise.
We will explain it first, okay?
For
So,
that question, there are different answers that could be the right ones because it all depends on the starting life cycle. By logic, I start where the spores got to the host. Those spores can come from different parts within the plant. They could come from the debris, from flowers that had been infected and remain there at the greenhouses, or from sclerosis that generate mycelium and eventually sporodio. Once those spores get to the roast and is recognized and acknowledge it, and then the environmental conditions are the right ones to develop botrytis that includes the free humidity within the leaf, relative humidity higher at than 93, and sun temperatures between 20 and 15 Celsius degrees. So the spores are going to populate. Once the spores are germinated, the fungus can also generate a specific mechanism that is called the appressarium, and it is a structure that is going to easier the nutrition and the penetration of the fungus within the vegetable tissue. Starting from there, the fungus keeps its invasive process, and once it goes on, we have the mycelial growth. That's what we get. Those cuts that you see, and the temperature is the right one. Those cuts or those lesions, they're going to generate new spores, and those spores are going to start a new disease cycle, and that's why we need to remove all the contaminated material from the greenhouse. That's why we have to remove it. Now let's go into the pool. Okay, that will be question number 3. I think that you skip it. Okay, now. Aquí and here. Okay, here I'm going to ask you another question. Okay. About the botrytis reproduction, is it's only sexual or asexual way?
for the English speakers, the question is, Botrytis cinerea only produces sexual spores. It is false or truth?
Okay, the right answer will be false, even though most of what we know about is true. The asexuated reproduction of this fungus, there is all evidence that there is sexual reproduction, which are as sport, and it's very difficult to find them in the cultivar. We can grow them at the lab, but we do know the existence of those spores, that can be reproduced sexually, even though the botrytis reproduction is mostly done through an asexuated process. It is a fungus that splits very easily, that has many nucleoids, and even though they are like semi-clones, they have a high adaptability and they can change, and they adapt very easily. So now let's go into another question. Which of these symptoms belongs to botrytis disease? And you're going to find 4 pictures plus 2 written answers.
Okay.
So, which of these symptoms are botrytis? There are 4 pictures, or you can guess all of the above or none of the above. Okay. And the correct answer is not E, it is F. None of those pictures are Botrytis. This is a very important point. So we have received flowers shipped from Colombia. We see spots. We culture those spots and identify the organism that causes those spots. And frequently, these are not Botrytis symptoms. So, for example, the top is Alternaria, Epicoccum, Penicillium, Cladosporium. The message is you can't tell the difference between Botrytis and these when the spots are small. The only way to really tell the difference is to put these petals into a humid chamber and see sporulation. To see the spore formation is really the only way to differentiate some of these symptoms. The penicillium, you do see some green eventually, but when the infection initially occurs, these organisms all look very, very similar. And this is one of the challenges we face. We ship flowers, and customers may make claims that you have Botrytis, and well, there may be spots, but it may not be Botrytis that you're dealing with. So, as an example, can we use the data? Yeah, so this is Botrytis. Before a sporulation, it looks the same as all of those other symptoms. Yeah. So this is a percent or incidence of the spores that we collect from flowers that we have received. And Botrytis is the most common one. Clearly, it is maybe 75% of the spores roughly, or infections that we see are Botrytis, but that means 25% are not. Okay? They can be Alternaria, Penicillium, Cladosporium, other things. Okay? So, we can do the next one in the survey. We have another picture of what I would call bent neck, with the flower nods off to the side, and you have an infection at the base of the flower at the top of the stem. And the question is this infection Botrytis? True or false? Or this, it's not a question. It's the statement. This is a symptom of Botrytis. True or false? Oh, you could not do it. All right, everybody made that. So the question is, will we try to fool you 2 times in a row? Huh? And the answer is false. This is not Botrytis either. Okay? And Melissa will explain.
Okay, this is a symptom of a bend down neck. Okay, okay, could be able to try this, but it could not be. This neck bending happens due to a physiological symptom. You know, when the humid is high, and and that's constantly once they are removed from that condition, so they are turn turgency, and once they lose turgency, and within the stem due to the weight, they are going to tilt the head. You know, they are going to bend the neck due to the lack of humidity. Now it happens that we have a botrytis infection at the base of the neck, and that where it generates the flower will collapse due to that, and when we place it at the humid chamber, we see that the botrytis is there. The symptom that we showed you at the pool belongs to the sclerotinian disease. Sclerotinian is a close cousin of the botrytis, and it is differentiated because it doesn't produce a spore. It doesn't produce spores. It produces an sclerosis that we can, when they are submitted to high humidity through many days, along many days. Now we're going to go back to the poll, and
The question here is, which of these tissues has the greatest potential to develop botrytis spores? The first is flowers. The second is stems that have been harvested that stay in the crop. And the third is leaves. And the fourth is floral debris. So the question in English
is the question in English is which one of these tissues has more potential for development of spores of botrytis? And the first option is flowers. The second option is stems in the greenhouse. The third option is leaves. And the fourth option is plant debris at the base of the beds.
Okay. And the correct answer is A. This is our opinion. So the flower petal tissue is the most susceptible to botrytis infection. So if you have flowers or petals on roses that fall to the canopy, that will be the first tissue that will be infected, clearly. You have a lot of debris in the bottom of the canopy. For the most part, it's not that susceptible to botrytis infection because it's mostly leaf tissue. And leaves on roses are quite resistant to botrytis. The second most common, and that's what you see here, leaves are very low incidence of botrytis. When we receive flowers from Colombia, we assess the leaves and rarely, if ever, find botrytis infections on the leaves. So when the leaves fall down here, they are not that susceptible to botrytis infection. The stems below the cut are also a location with a high frequency of botrytis infection and sporulation. Those stems, when the cut is made, you have the tissue dies down to the lower node below the stem, and that decaying tissue is quite susceptible to botrytis infection. And so we don't often see dead flowers if we're growing in a clean operation. We don't have flowers on the floor, hopefully, or flowers on the soil. And so we don't see infections on flowers in the greenhouse too often because the greenhouses are clean. But this wounded stem is a very common location for spores to form and be dispersed throughout the greenhouse. Okay.
So let's go back to the poll. And the question is, in which of these petals is there a higher propensity for the development of botrytis? The first picture is indicating the external petals. The second is intermediate petals. And the third, the internal petals.
For the English speakers,
so you have petals on the outer petals, on the outside of the flower, the middle petals, the ones that are just inside the outer petals, and the inner petals, the small ones in the center. Which of those are the most likely to see botrytis infections on? Outer, middle, inner. This is also a tricky one. In my opinion, and what we see on roses, we've looked at many, many roses, the middle, the inner layer, the middle layer here of petals are the most susceptible, or the most, that's not true, not the most susceptible, the most likely to have infections. Why? The outer petals have been exposed to fungicide applications. Because the rose has 3 outer petals that cover the bud, and when we are spraying fungicides, we have contact with those outer petals quite, every week. And so those petals get good protection. But as the flower opens in the last week before harvest, the middle petals become exposed. And then a spore lands on those middle petals, and those petals do not have a lot of fungicide to protect them. And so we often see that in the vase or in the humid chamber, the outer petals look quite good, but that next row of petals inside are the first ones that show infection. The very the inner petals are susceptible to spores infecting them, and they don't have fungicides applied to them, but they are also not exposed to the spores very much because they're small and inside the bud, and they open up and are visible in the vase, not in the greenhouse. And so spores don't have the opportunity to land on those inner petals very easily. Okay?
Now, once again, we're going to the poll, but we're going to move a little bit to the monitoring side. So the question here is, with which of these strategies for monitoring can you confirm the presence of botrytis? The alternatives are Petri boxes in the greenhouse, humid chambers with the infected tissue, or revision of lesions in the greenhouse.
The question in English is with which one of these monitoring strategies you can confirm the presence of botrytis. And the options are Petri dishes in the greenhouses, humid chambers with tissue, or looking for lesions on the tissue in the greenhouses.
Listo. So yes, everybody agrees on this one. The most coherent form and easiest of confirming the presence is with humid chambers. With the Petri boxes, we can also confirm the presence of the spores in the greenhouse. But when we put these Petri boxes, many times, the majority, it's not just spores of botrytis, but other contaminations. And also a lot of contaminants that not necessarily are going to result in some type of disease. But when we put these petals with the spots in a humid chamber, and these humid chambers sporulate and give us botrytis, then yes, we can say that's the issue that we have. And the monitoring in the greenhouse is good to help us detect those spots, but it should be accompanied by those humid chambers. I'm not saying that the monitoring in the field doesn't work, but we need to determine where those spots are, independent if it's botrytis, alternaria, epicoccus. We want to get rid of it and get it out of the greenhouse because we don't want to leave this pathogen source. But it's important to confirm with those humidity chambers. Now we'll go to the next questions. Which is, how many days do the infected petals with botrytis take to sporulate in a humidity chamber? More or less 22 degrees centigrade.
And the question in English is how many days infected petals with botrytis take to sporulate in a humid chamber? And the possible answers are 1 to 2 days, 3 to 6, and 7 to 10 days.
Very good. You've got it extremely clear. Yes, effectively, in the conditions that I described, this is the time more or less that the sporulation takes. From day 3, we can start to see that some of the petals begin to sporulate, but we're going to have a population of some petals that are completely sporulated, more or less around 6 days in general. Now, we're going to pass to talk about cultural practices. And for that, we're going to go back to our poll. So here the question is, which of these activities is not an activity that generates a high dispersion of spores? And the options are harvest, cleaning of the floor debris, sweeping between the beds, and changing the refreshment areas in the greenhouse. And the correct answer is the sweep between the flower beds. And I'm not telling you that it doesn't generate, but it's not the one that generates the most. In general, everything generates movement, but it's not one of the ones that generates the most. We conducted a study where we had 2 spore collectors in 2 commercial greenhouses in the savannas in Bogota. And these collectors took samples that had spores, and these samples were taken daily. And we had a record of all the activities that were carried out in the greenhouses in each of these days that we took samples. So in correlating this data, we found out that these that I'm showing you here with other practices are the practices that cause more movement of spores. And here, for example, I want to make a clear note. In this practice of getting rid of the refreshment area of the greenhouses, it's a practice that's very delicate, because not just is it going to move spores, but in having fresh water in these greenhouses, we're going to have a microclimate that could be favorite for the germination of spores. So not just do we move them, but we're also facilitating the process of germination. So another of the practices that we saw that had high incidence of the movement of spores is the cutting and removing of dry matter, like the recollection of this plant debris. And this is what you see in the picture. It's concentrated in the beds. And it's this sweep that we do in a horizontal fashion. And this was highly related with a high movement of spores. Now, and the removing of contaminated matter in both cases of botrytis. And it also showed some tendencies for the movement of spores. Now, once again, we'll go back to the poll. And the question here is, how probable is it to find spores in the cold room?
And the question here is, how probable, how likely is to find a botrytis spores in the cold room?
So for this experiment, what we did was we had spore collectors, and we put the spore collector in a cold room and let it run for several weeks to collect botrytis spores. And so the question is, how many botrytis spores did we collect? A little bit or a lot? And the answer is, well, a little bit. Why? Okay. Why? So the flowers that are in the cooler have just been cleaned. There's no botrytis infections, and so there's no potential to produce spores. So thus we did a really bad job cleaning the flowers in the packing room. So in the cooler, did we ever find any spores? 1 or 2? We cannot collect spores in the cooler at all because they're not there. There's no debris. The floors are clean. There's no material plant material that is infected, that is producing spores, and that's what you have to have. And it's also a low temperature, so you're not expecting even if you did have tissue in there, you would not expect it to grow very quickly, and the stems are not in the cooler for that long. So the answer is no, we don't worry about botrytis in the cooler.
Now we'll pass to talk about the climate. And now here the question is, in which of these temperatures is there a greater risk in developing botrytis? And the possible answers are between 10 and 14, 15 and 25, and 26 and 30 degrees.
The question in English is, in which one of these temperatures there is a higher risk for development of botrytis? And the possible answers are 10 to 14, 15 to 25, and 26 to 30.
The optimal temperature for botrytis to grow is 20 Celsius. So this is a model to predict botrytis infection on strawberries. So this is used in the States as a way to anticipate infection in the field and to know how to time your fungicide applications in field production of strawberries. And what you see is temperature on the x-axis, and the optimal temperature for infection risk is 20. But you actually do have risk at temperatures from 10 to 30, but optimally 20. And then the other factor that we would use to predict botrytis infection is the number of hours in the day, with the 24-hour period, number of hours in a 24-hour day that the plants are wet, where there is condensation on the tissue, or the humidity is over 93% relative humidity. And so what you see here, the blue line, is where we have 12 hours of continual leaf wetness every day, or during this particular day. 15 hours, 18, 20, 24. So you see, as the number of hours that the tissue is wet, the risk for infection is much, much higher. So if you have 20 degrees and leaves have condensation, or excuse me, flowers have condensation on them for 24 hours, it's like 100% chance that you are going to have botrytis infection if spores are on that tissue. So one of our goals for botrytis management is trying to minimize the amount of condensation, which is difficult to do when our night temperatures are quite cold. Yeah.
For this, we're going to answer another question. And the question is, in which of these situations is there a greater risk in developing botrytis? And the possible answers are air temperature of 10 degrees and a dew point of 12. The answer is air temperature at 10 degrees, dew point of 10. And the third option is temperature 10 degrees and dew point of 8.
The question in English is, in which ones of these conditions is more likely to get botrytis risk? And the possible options are 10 degrees Celsius and dew point of 12. The second option is 10 degrees Celsius, dew point of 10. And the third option is 10 degrees Celsius, dew point of 8.
Well done. So when the air temperature gets below the dew point is when we are in risk of condensation. So this is a situation where fans in the greenhouse can be very beneficial because it will help to dry the canopy so that the tissues, the plants are staying wet for fewer hours during the night. So when morning comes, the plants are drying off and not having free water on the surfaces.
Superficie sobre el tejido. Okay, now let's talk about a little bit about fungicides, the handling, how to manage fungicides. And to do that, we are going to ask you another question. Here the question is what fungicide may present a higher risk to develop resistance?
So the question is to which one of these fungicides there is more risk of resistance development, and the possible answers are multi-site fungicides or single-site fungicides.
Entonces, that's right. Okay, that's okay with the fungicide specific site. There is fungicides that add in a single spot, a single enzyme, and that's why it's easier that the fungus to generate mutations or changes that will get over this type of action. And the multi-site had many action sites, so the fungus is very difficult for the fungus to adapt or to develop resistance to the fungicide. Additional, the specific site fungicides, they are called fungicides with a higher efficiency as long as we don't have any resistance. And additionally, on the top of everything, the efficiency is deemed moderated in the multi-site fungicides. Now we are going to go into our next question. And the question here will be, which is the action method for the specific site fungicide? So one will be systemic that go into the tissue and by contact without any tissue penetration.
In English, what is the mode of action for site-specific fungicides? So are they just protecting the tissue by staying on the surface, or are they systemic locally, penetrating the surface and controlling the mycelial growth? And A is correct, they are locally systemic, and this is the difference, one of the differences between multi-site products. Multi-site products are protectants. They are on the surface, they do not penetrate the tissue, and we do not want them to penetrate the tissue because they tend to be phytotoxic if they actually go past the cuticle into the leaf tissue or petal tissue. So we actually want them to stay on the surface, and their mode of action is to prevent spore germination. Hence, their value is as a protectant, because once that spore is germinated and penetrated the tissue, multi-site products are not effective. Site-specific products are effective after spore germination because they actually penetrate the tissue and can influence mycelial growth in the tissue.
Vamos a la encuesta otra vez.
And here the question will be, which of these fungicides that had a FRAC7 code will be more probably that Botrytis show resistance? We have boscalid, hexamine, isotaminide, and fluoropiran.
So we have several FRAC7 fungicides that we can use, and they are not all equal in preventing resistance, or having resistance to them. Some FRAC7s have a high level or moderate level of resistance, and others are still quite effective. Which one of those four has the highest probability of fungicide resistance occurring? And it looks like we did very well on that one also. Boscalid there's much more resistance in the industry to boscolid than the other frac 7 products. And so, yes, question, Diego.
Why Boscolid more?
I'll let Melissa. Melissa gets the tough questions. I get the easy one, okay?
Okay, there are many things there. In general, when we talk about fracs, when we talk about frac codes, you said I had to do the rotations by frac code because they group the fungicides that have the same action mode, but within the frac 7s, like within the frac 3, there are some specific differences that are going to give me some resistance. Not all mutations will give me resistance to the same fungicides. Boscoli is the most doable because it has been in the market for a long time. So the fungus had been under more pressure. And here, for instance, let me show you many of the products that we have assessed. And as you can see, up to this point here where we did the assessment, this new molecular P-methoxy is shown lower resistant because the spores has never been exposed to this fungus. We did this before the applicating was done. And for instance, in the case of the fracts 1 and 2 that have a high resistance. So this is a condition that not only happen with roses, but we can see different crops and many ornamental crops in the United States. These two molecules, leprobion and morganate, they show a lot of resistance. And here I would like to highlight something that is important and relevant. This is data that we got from a commercial farm in an assessment that was done through a period of 6 months. But please do not leave here with a message that that will always happen because those conditions and the resistant conditions are going to change accordingly to the management of the cultivar and the molecules that are used within each farm. So everything is going to change. So here, I can imagine that most of you do efficiency test, internal, external, or you may send or you submit you submit your samples, and there are some companies that do this type of assessment, and that's important. It changes a lot up, so it is important to know how to interpret the results. So as I said before, this is a snapshot of what is happening at the moment that the efficiency tests are conducted, that we were able to see that within a month this efficiency and the snapshot is going to be totally different. So the more assessments you conduct through time, it will be more accurate, the decision making process regarding efficiency. So when you have, for instance, when you know that your isolated population has a resistance less than 20% to a molecular, the efficiency is going to be effective if you apply it individually. Okay, and you are going to reach that 20. When we move between 35% and 50% of the isolations that present resistance, we are going to increase the risk of the spores that are going to survive. And at that point, it is important to come up with mixes of the multicide plus a protectant, because the fungus spores that are going to survive, because they have more resistance or are more sensible. So the protectant is going to avoid that the spores will germinate, and we are going to keep low the population. And if we had a density higher than 50% in the development of resistance, so the best thing to do is get rid of it. Okay, now let's go back to the pool. After stopping using a fungicide for a long period of time, is always possible? Is it possible to recover the efficiency of that fungicide?
The question in English is after a long period of ceasing a fungicide application, is always possible to recover the effectiveness of that fungicide? And the possible answers are false or true.
That was a tricky question because always and not always. Okay, usually the effectively we can say that effectively we can recover some molecules if we stop using them through time. But this is only through if the development of that resistance is linked directly to biological efficiency or what we call fitness. So this is the fungus capacity to survive and reproduce. In many occasions, that resistance and survival takes, I mean, it's going to take something away from the fungus, virulence or some capabilities. So that is going to pass a tap on the fungus, all right? So when we stop using the fungicide after a period of time, the spores population that do not have any resistance, even though they have that plus of biological efficiency, they are going to start taking location over the resistant population, and eventually we can recover the efficiency. But nevertheless, so that the development of the fungicide it starts with a mutation that do not carries out any biological charge. So for the Botrytis to develop resistance to the nitrite butane is not sacrificing, and you can use this for 200 years. The population is never going to recover. Okay, so you can use it and nothing is going to happen. Now we are going to talk about these management alternatives, and we are going to talk about calcium. And now we go back to the poem, and the question will be. Okay, more than a question, it is an state. I mean, I need you to arrange the ideas. Okay, arrange the tissue from higher to a lower concentration of calcium, and we have
the
this.
English is organize these tissues from the highest to the lowest calcium concentration, and the possible answers are stems, leaves, sepals, or petals. Yeah.
So we receive roses and break the tissue down and send that tissue for analysis. And what you see is dramatically different levels of calcium in different tissues in the stem, or within a cut flower. So the petals, inner, middle, and outer petals, all have very, very low levels of calcium, 0.1, 0.2%, very low. Stems also are relatively low, which is surprising. I think stem was the number one answer. And stems that have low concentrations of calcium. The stamens are a little bit higher. Sepals are very high, and leaves are very high. So what you see is that tissue that is transpiring a lot of water has the opportunity to accumulate a lot of calcium. And so rapidly developing tissues and tissues that don't transpire water very much, like petals, and stems don't transpire much, don't have an opportunity to accumulate very high levels of calcium. So the levels of calcium in the leaves can be 10, 15, 20%, 20 times higher than what is in the petals. And this is one of the reasons that we expect or theorize that petals are so susceptible to botrytis, because you see a completely inverse relationship between calcium concentration and severity of botrytis infection. So we have the most botrytis infection in the petals, and then the stems, and then the stamens and sepals, and leaf. We see very, very little risk of infection. So it's a perfectly inverse relationship between calcium concentration and infection rates.
Vamos de vuelta a las preguntas.
So this underscores the importance of calcium management for botrytis.
Here the question will be, which is the best way to provide calcium to the leaves? The first one is a foliar application.
So if we want to improve the level of calcium in our tissues, what is the best way of doing it? No, that's not what is the best. Is that what we asked? The most effective. What is the most effective? Fertigation, applying the calcium in the irrigation solution, or spray. Or the other end, there's three answers? Just two? Okay. Oh, this is just for the, okay, thank you. I have to clarify, we're only talking about calcium in the leaves, okay? Sorry if my Spanish is not that good. And well, it's actually terrible. So, but it's getting better. Okay, so the answer is both. Both are effective. Fertigation is a very good way to accumulate calcium in leaves because the leaves are transpiring. So a lot of water is coming up through the leaves, and calcium is transported with the flow of water in the tissue. Okay, so we have to have water flow to get calcium uptake. Plants do not actively take calcium up. We have to have transpiration to get calcium uptake. Sprays are also effective. If we spray leaves, we will get more calcium in those leaves. So if we're trying to accumulate more calcium in the leaf tissue, we have two techniques and they both work. Okay?
Okay, let's go back again to the poll. Now we are going to stop talking about leaves and we are going to talk about the petals. Okay, so the question here is, which is the best way to apply calcium to the petals? Here we have three questions. What the first one is foliar aspersions, and then is dipping in the post-harvesting.
So we're looking at tissue concentration of calcium in the petals, and we have three options: fertigation, spray, or dipping in the post-harvest environment. Okay? And the answer is C. C. We can get very good accumulation of calcium in flower petal tissue with a very simple, fast dip of the flowers before shipping. And I think Melissa will show us data.
Okay, what I'm showing you here is the results on the assessment that we conducted through 20 weeks of applying calcium once a week at different concentrations, including 500, 1,000 per million. Foliar parts per million. In the first graph, we see the relation between the botrytis sensibility and each one of these applications, and we can see that the applications at 5 and 10,000 parts per million managed to gain lowering the severity of the botrytis. But when we go on and we analyze the tissue, the applications that were focused on the petals increase the concentration in the leaves, but in the petals, that is this line here, we don't see any change in the calcium concentration with regards to the petals. Now, we conducted some application in some different post-harvesting of 15 seconds of the whole bush the whole. So because the solution is going to go into the internal petals, we lower the severity of the disease. We could see that, but at the same time, we see that there is an increase of calcium within the petals, and at mil and 2 parts per million, 1,000 and 2,000 parts per million. Okay, now we can see that the leaves was not the objective of this application, but they were benefited because the
So after a dip application, we are accumulating more calcium. It's still a very low number. This is 0.2, 0.3. So we're doubling, almost going from 0.15 up to 0.3% calcium. It's still not a lot of calcium, but that's a 100% increase in the calcium in the tissue. So we see a big response. You see a response in the leaves also, but the leaves already have fairly high levels of calcium. So it's really an important technique for accumulating calcium in flower petal tissue. Diego?
It's important to do together with calcium and fungicide. Because if you put calcium and water,
Yeah.
you need to put something to protect the flower.
So the question is, do you have to mix the calcium with a fungicide? Because if we just put the calcium in with water, then we're wetting the petals and maybe making them more susceptible to Botrytis. We didn't answer that name on it. You.
in this case, we did it with a company that choose not to use fungicides in the post harvesting. And in this case, our commercial control at this point was the hydrogen peroxide. It was a molecule that was being used in the post harvesting. And, lately we had been creating some mixes, and later I am going to explain a little bit with different molecules. But the truth, we haven't tried out with fungicides, but I believe that this could be an alternative. And we need to see internally what are the dosages to see which is the toxicity and how we can apply it. Y él está preguntando de la compatibilidad. Okay. Sí, okay. Okay, he's also asking about the compatibility between fungicides and calcium. And for some growers, we know that there are some mixes that do work and some others do not work. Okay, we have seen negative effects of that combination that you were talking about. But this sclera is an issue by itself if you apply it. I don't have the answer based on the data that I have because we have never done it. That will be that like internal. That's something internal that you will have to do. So my advice to you is to take all the mixes that you use in the post-harvesting and do mixtures at different concentrations with different cultivars, and from there you can assess, which ones are effective.
No vale la pena aplicar calcio, foliar, recorte corte crisantemo.
Okay, the question was based on the data that we have, are we going to apply foliar calcium? Okay, pre-cuts.
Es una práctica, digamos, que es muy generalizada en la zona, aplicar calcio, cada que hay un problema de que se aplique calcio. Pues llega uno al extremo a aplicar 3 veces calcio en esa edad de
Okay, what I think about.
So, okay. So the question is it really a benefit to apply calcium in the greenhouses during the production until the post-harvest? Because we don't see an increase in the calcium concentration in the tissue. And it has been believed one of the first practices that you have to do for botrytis management.
Oh. Is it worth it to do this to apply this throughout the whole production process, even in the post-harvesting, even though we don't see any benefits in the concentration of the tissue. And it is deemed as one of the first practices once we to determine botrytis is there present. Pásale el micro. Okay, when I'm asking you, with the results that you have gained, with all that data that you have compiled, is it worth it, continue with this practice that we have here in our region? With chrysanthemum, is that we applied at week 7 and 8 when the bottom had blossomed, and until the week 10 there when we start cutting, we apply calcium 3 times, even though we know there is botrytis there. Okay, I mean, is it worth it to do that?
Okay, so this data was on roses, but if we didn't see an increase in the concentration of calcium in the tissue, there is a minimal decrease. It's not a huge decrease, but it could help in the management. We'd have to look at the concentrations, see what concentrations it's being applied at. All right, if the other technicians can correct me, but the thing here is to have between 200 to 350 ppm maximum of concentration. I mean, sometimes you reach 500, but it's very strange because of fear of toxicity.
I didn't get that. If you want me to answer.
He said that he was presenting with normal applications of calcium.
Yeah.
So can we see what the botrytis, but the lack of what we found, we recommend those practices.
Yeah, we think calcium applications are valuable during production. Yeah. Not just as a dip. Yeah. But it goes into what you're going to talk about with so you want to mention that.
So I'm going to show you a little bit of the evaluation we've done, where we don't just see the direct effect of calcium on the level of accumulation in the tissue, but there's another effect. So I think up to a certain point, these applications can help with that other indirect effect. So here, as I was showing you, we had concentrations of 500 or 1500 parts per million, which is really high, but the range between 500 and 1000 is where we saw that effect. When we started to go above this, then we had a phytotoxicity risk. But I repeat, we do see a small benefit, and it could be associated to what I'm going to show you. What source of calcium were you using in the study? Calcium chloride. Why do we say calcium chloride? Because in evaluations prior that were done with petunias, we evaluated various molecules, and among those molecules, calcium nitrate and calcium chloride were the molecules that presented the least amount of phytotoxicity and a high effectiveness. And that's why we went through the chloride instead of nitrate, because when we're talking about the management of botrytis, we're not interested in increasing the nitrogen concentration, because this favors succulent tissue, which in the end is really attractive for botrytis. Any other questions? And this that I was showing you here, this concentration that is one with the dips is reflected, that increase in a strengthening of the tissue. How do we measure this? So these petals that were submitted to dip, we made the dip, and 24 hours after that dip, and 7 days after that dip, we measured through a machine that's called a texturometer the strength necessary to break that tissue. Once we measured this, we saw that the treatments from 0 to 1,000 didn't manage, well, the 1,000 didn't manage to increase that strength of the tissue. Even so, with the treatment of 2,000 ppm, that increase of calcium that we saw in the prior graph, we saw it reflected in a greater strength of the tissue, which means we need a greater load to break that tissue. So we do obtain a stronger tissue when we make these applications. So let's go back to the poll. All right, Melissa, in the lab, this is measured percentage from ash, or the levels are taken how? In fact, we made 2 evaluations of tissue. One is on a scale of ash with ICP technique that's current. And then we did another evaluation that, although I'm not sure it here, has basically the same data. And it's an evaluation of a spectroscope X-rays, where what we do is we look at the transversal piece of the tissue, if there's calcium, how it moves from the epidermis to the internal part of the tissue. When we carried out these X-ray evaluations, we could see that, in fact, that greater accumulation of calcium, we didn't just see it in the epidermis, but the mesophilus also increased. The calcium, so it is moving within the leaf with these applications. So the calcium, is it associated? It's in the walls. I mean, this is what we don't know. What's it measuring? So I don't know if with X-rays that you measure that calcium at the end, where does it go? Where does it stay? Well, the technique doesn't allow us to know in which of the organelles is where the accumulation is. But the theory tells us that what isn't in the cell wall goes to the outer structure and in the chloroplast. But the greatest accumulation is vacuoles, because the cytosolic calcium is very low because of something that I'm talking about in a minute, because it works as a strategy of understanding for some external signals that the plant receives. So those small changes that happen in the cytosol can give us as a result some answers in the plant. So in general, that calcium, extra calcium, more than likely goes to the vacuoles, and it sends messages for the plant when it requires it. Here, we have another question, which is the CAPC helps the effectiveness post-harvest of calcium, and the proper answers are true or false.
And the question is, the coadjuvant capsule helps the assimilation of calcium, and the possible answers are true or false.
Okay. Our data show that the answer is false. It's not intuitive. When we add the capsule, we seem to get better coverage and would expect to see more calcium uptake. So we've made many measurements, and I think we have the data here to show. Here's the dip or immersion treatment, and with no calcium, 1,000, 2,000 parts per million, and then we have without capsule, with capsule. And you can see there's no difference in the amount of calcium that's taken up by the plant, whether we have capsule, coadjuvant, or not. This was a surprise to us also.
The prior is what we saw in terms of severity. So we didn't see an improvement in the effectiveness. And here we see that we didn't either. I mean, you can see here that in the continuous black lines and the dotted line, there isn't a difference between them and those with capsule and without capsule. Let's go to the last question. And the question is, the applications of calcium don't have an inductive result of resistance, and the possible answers are true or false.
In English is calcium applications have no effect inducing resistance in the plant, and the possible answers are true or false.
False. So this is Melissa's newest work. We've always kind of assumed the main mode of action for calcium was to build stronger cell walls so that botrytis or fungi doesn't penetrate the cell wall as well. Melissa's newest work shows that, and she's going to show you a few slides that initially look very complicated, but she will explain it well. It shows how calcium actually helps to induce the plant to be more resistant to botrytis infection.
Okay, so I'm going to try to do this as digestible as possible because up to the moment it's a little complex. But basically, what we did in this evaluation was the same dips that we talked about. And those dips we did just calcium treatments to see what happened. Just inoculation, see what happens to the plant, and really the flower. And here I think it's very important that we all be on the same page that these are cut flowers that received a treatment post-harvest, and this is what happens to this cut flower. And we'll see the interaction between calcium and botrytis. Now, in this first one, I want to show you what happens only when we apply calcium. So we can see these that we see here are different metabolic routes that had a significant difference when we carried out this application. We can see others that increase and some that diminish, but in general, we are moving the needle for these metabolic routes for the plant. Here, I'd like to highlight, here we have the top of the metabolites that are individual, that belong to each of these routes. And here we can see that of those top 10, we have 3 that are from the purinas, and some that go up, others that shoot down, and others that will give us different reactions. Here's one that I think is very important to highlight, which is the phenylene, tyrosine, and tryptophan. And you can see that 2 of these molecules that we found in the top 10 are part of this route. So why is this route important? Because it's a route that has been related with an answer of making agile this induction to resistance. This phenylene, for example, has an effect where it makes it quicker, the answer, once we detect the pathogen. So what it does is it turns on the alarms of the plant so it can answer much easier. Here, we can see the answer of the flower to the inoculation with botrytis, just inoculation with botrytis. Here you can see that the botrytis is doing a huge amount of things in the flower. It's manipulating a huge amount of metabolic routes. Some of them are results of the flower trying to defend itself, as we can see with the route of alpha-linoic. And here in the molecules, the top molecules, we can see hasmonic acid that historically has been associated as one of the first defense routes of the plant. So for fungus that are necrotic, just like botrytis. So we can also see other routes that historically have been related with the production of antimicrobial compounds like brasostenoids. In this case, the plant is trying to defend itself. And we also see something that I think that we should highlight. And this is that the infection of botrytis is making some changes in the sugars of the plant. makes that the plant have a greater production of sucrose and of starches and other compounds like fructose and mannose, which are other types of sugars that aren't as favorable for the production of botrytis. They actually go down. Now I want to show you what happens when we apply calcium and 24 hours later inoculate the plant with botrytis. So in this case, we're going to see routes that diminish and routes that increase. And I want to concentrate a little bit on these routes that increase because this is where we think that there's a key point for the management of the resistance. So here we can see that there are 3 compounds of that top 10 that make part of 3 metabolic routes that are important. One of those routes is the productions of cutin, suberin, and waxes. And what happens when we penetrate, the botrytis generates a rupture of the cuticle. And this, what it's telling us is that the plant tries to superimpose that rupture, making a kind of scarring of the tissue. Additionally, we also have the routes of fatty acids, saturated and not saturated, which are also associated to bilipic membrane, which is part of the cells. And what I'd like to highlight most here is that we see that 2 molecules are related with metabolism of the sphingolites. So one of these is phytosphingoside, which is a molecule that has a direct effect in the the hypersensitive response of the plant. It could be associated that the flower take the decision of killing a cell to diminish and stop the invasion of botrytis. Finally, we also have other defense routes like phenylpropanols expressed with these applications of calcium. Now, the message I want to leave here, and I know it's extremely complex, is that with the applications of calcium, not only can we get a direct effect with a greater accumulation, but we're also turning on other things in the plant that could be related with the defense answers. Now we're going to leave it a little aside, the calcium, and talk about natamycin. Yes, Marco? With this that you're saying of resistance inductors in the market, they're offering amino acids. This morning we were talking about amino acids, osines, toconines. Now, this type of substances that are applied directly, wouldn't this be favorable for these activation of these routes if they're applied directly, including vitamins? We've talked about vitamins directly. And I think commercially there are offers of this. I mean, do you think these applications would be favorable for the levels of calcium in the tissue? I think these applications of these molecules independently could have an effect in the response of the plant for botrytis, but I don't think it's going to favor the accumulation of calcium. Maybe a little the phenylalanine. There's evidence that there's certain correlation, but the other metabolic routes, no. Now, I think there's a lot of potential in these molecules. And if I'm not wrong, in Ciflor, they talked about some of these vitamins and potential. I can't say yes or no because I don't have data in respect to this, but I do know there's evidence that these vitamins, some like thiamine, riboflavin, can do that. And compounds like phenylalanine could help induce resistance. But I think we'd have to look at it. Okay. Now we'll pass to natamycin. Natamycin is an antifungal compound that is produced as a result of fermentation of a bacteria, which are streptospin. They receive their name from Streptomycin natalis, but it can also be produced by other streptomyces. Now, natamycin is a product that's been used since 1967 as a preservative for food, especially in the dairy products and meat products that have been processed. And this is one that unites to ergosol and it's fundamental for the development of fungus. Now, on blocking this ergosol, it stops a production and this will eventually kill the fungus. This natamycin can be found registered in the United States for the management of different agricultural products, and the registration includes its use in seedlings for the transplant of strawberries and the treatment of other plants. And there you can see that none of those registered uses includes the use in the field. Why? Because natamycin is sensible to ultraviolet light. So it finds opportunity is under controlled conditions. So for the moment, there's no report of development to resistance to natamycin. This is why we believe that there's a certain potential for the use of the molecule in.
So here I'm going to show you the results of some assessments that we have done. And I'm going to show you two different scenarios. In the first scenario, we apply natamycin by itself, 125, 500 parts per million, calcium by itself, 2,000 parts per million, and combinations of natamycin with calcium at 10,000 parts per million. The applications in 24 hours after we applied, we did conducted the Botrytis, the Botrytis. We took this to the chamber and we saw how the Botrytis has been. What was the behavior when we apply this combination 24 hours before? The results are great, but there's not too much difference when we did it single and then we combine it, and the difference is not too much. We use fungicide, fluoricide as a control because we knew that it was going to be a very effective fungicide, and so that was our control, and the results were not too different. And here we did basically the same, but the natamycin was applied 7 days before the inoculation, and in this case we did the applications of dynamic design at the post-harvesting of the producers. We did it by dipping, and here we could see that after 7 days between the application of the natamycin and the inoculation with Botrytis, the Botrytis is not going to be high. And here with the treatment of 500 parts with part, there is lowering in the severity of the Botrytis, but it's not significant. So here the calcium is constant, but we see the combination of natamycin at 500 parts per million and calcium at 2,000 parts per million. The mixture, something in the molecule prevents the wearing down of the natamycin, and then you see the accumulated progress when we go at the higher part, the 750 parts of natamycin and 2,000 parts of natamycin. We start going into toxicity. We did this with the samples of 3 different companies, and in one of these companies we started seeing this phytotoxicity problem, and we can compare this program with the non-physical. We didn't see the problem there when we compare it with a different outcome. So this is a visual view of what I was talking about, with the graph where we can see that natamycin by itself doesn't do anything. It doesn't do anything for the plant when you apply it 7 days before the inoculation, but we can see an improvement in the development. So we lowered the symptomatology once we go in this direction. Here, I want to show you a little bit of what what another of my colleagues did, Gabriela Caledonia. She assessed some biological products in the post-harvesting, and here she shows different products that she assessed under the dipping conditions. Okay, under this system, I'm not saying that this product do not work. I'm not saying that. I'm saying that that work when under this system. So most products didn't do anything, to prevent here. Cilion, this is the commercial name for natamycin, and plus another calcium molecule. Here and here, this is the control sample. They did have effective, an effective.
She's next. I think this is our last slide. We'll wrap this thing up quickly, but a very interesting point when we start looking at biologicals is how effective they are. We don't expect them to be as effective as traditional chemical fungicides, but we hope they're close. With this experiment, as Melissa had shown you before, natamycina was very effective if spores were being applied a day after the application of the natamycin. But if there were 7 days between the application of the treatment, natamycin, and the application of the spores, the effectiveness was not very good. And so we did another experiment that proved that point. And so, again, we have severity, and this is the commercial control, Verivis prime. And you can see we have very good efficacy, whether it's 1, 3, 5, or 7 days between treatment and inoculation. So that product worked very well, even if the spores are landing on the petals 7 days after the treatment was made. If we look at natamycina here, you see a trend upward as the number of days between application and inoculation occur. Hello? Okay. So we have good efficacy if the spores are applied 1 day after treatment, and some efficacy after 3 days between treatment and application, but inoculation. But at day 5 and 7, the effect has worn off. It is no longer effective. This is demonstrating that the compound, natamycina, is breaking down. It is losing efficacy in a very short period of time, which makes it not as good of a product as we were initially hoping, but it does show that it can work. It just does not work for a long time. Interestingly enough, Polyoxin D, trade name Affirm, has an exact opposite response. If we spray Affirm on the plants 1 day before inoculation, we get no effect. It looks the same as the control. But after 7 days, it's actually, the tissue is much more resistant to the Botrytis infection. You see the yellow line coming down very significantly. This is an example of induced resistance, where the compound is turning on defense mechanisms within the plant so that the plant is resisting Botrytis more and more with each day after application. Yeah. So one could then hypothesize, if we mix a product like this with a product like this, you actually have good control for 7 days. One product works early, the other product works a little bit later. And so we do a treatment like this in a post-harvest environment, because natamycina is not going to be a greenhouse treatment. It breaks down with UV light very quickly. So it's only going to be post-harvest, but it only works for a few days. So perhaps a combination of those two might prove to be a very good post-harvest treatment.
How's your interpretation of the data of the control not inoculated in severity of trials?
Did you understand?
Okay, given the fact that our samples come from the field, it's very difficult to have a non-inoculated control because they come from latent diseases, but in this case we didn't apply anything. So the only thing that we applied was water in the control, and this is the standard development of these latent diseases. So what I gathered from here, on the control we have the spores that are there, and regardless of how many days have gone by between the plant getting disease and the exposition of an ideal environment for the development of the spore, the latent infections are going to be very successful as long as the environmental conditions are the right ones. So we're not going to see any changes in those latent diseases. They are going to be there. So we need to lower that natural percentage of the disease, and send it to control, just to see the efficiency of the inoculation. Okay. We haven't. Yeah. You're totally right. We haven't done it, but.
Questions? Marco?
Okay, going back to the resistant. Okay, and trying the resistant indicators. So we have been offered these algae extracts, sargastrophe, so some algae, Nascophyllum nodosa, that is being harvested back there in Canada. So I would like to know your opinion with regards to that type of product. This algae that they are sending from Canada. Okay, I'm going to be very honest. I don't have any experience with that kind of products, and I don't dare to say if they do work or not. That's the short answer. Okay, there's some evidence that they may work again on some fungus and including botrytis, but I don't have any evidence and if they work here in the flower production. So we'll have to look into it, but because we need to be working on different managing alternatives, but I cannot tell you if the product works or not. We don't have any experience.
Any other questions? It's been a very long afternoon.
Have you any knowledge about the interaction between UV light and botrytis?
Okay, I know that there's some type of UV light, the UV light B. Yeah, if I'm not mistaken. Okay, there's some effects in the lower of botrytis. Yeah, there has been. The question was, has there any effect of the UV light on the botrytis? I do know that there is an effect. She's saying there's some strawberries back in the Florida. They had some carriage that go through night, and they had shown some high efficiency to control botrytis. When we talk about flowers cut, the architecture of the plant is more complex, and we will need to assess different systems just to be able to be effective in the application of UV light. But we think that there is a high potential of using.
So the moment you have all been waiting for. The drum roll. So, first place, oh my gosh. What, is it this column? So we have two people that had perfect scores. My gosh. We have Juan Yaro Lopez. Please stand up. You, stand up. Second place, Reese's. But actually, it's tied for first place. Tied for first place. And the first one.
Luis Felipe Jaramillo.
Somehow there was some cheating going on there. Something fishy. Team effort. Okay, you've been a fantastic audience. Thank you for your time. If you have any questions, please come up and we can talk some more. It's been a pleasure speaking. Thank you. Thank you.